So we are starting with the tryptophan operon. Welcome back. We are starting with tryptophan operon. So here in tryptophan operon, so here the first one was metabolism of lactose. Here we are going to see an operon which helps in the synthesis of tryptophan inside the cell. That means the enzymes or the genes which are being which will be discussed here in the operon is causing the synthesis or synthesis of tryptophan. So tryptophan is an amino acid which the bacteria synthesizes by itself. So that it is biosynthesis of tryptophan. So we are going to see how the enzymes which are involved in biosynthesis of tryptophan, that means the synthesis of tryptophan inside the cell, inside the bacterial cell, how that is controlled, how the enzymes which causes the synthesis of tryptophan is controlled inside the bacterial cell. So make it clear the differences between lactose operon and tryptophan operon. Lactose operon was degradation of lactose into glucose and the enzymes which are involved in degradation of lactose into glucose and galactose. Here we are discussing an operon which is a biosynthetic pathway. That means here we are going to see the enzymes which are involved, or the operon in which you have the enzymes which are involved in the synthesis of tryptophan inside the bacterial cell. So here the operon is activated whenever, so that is obvious, whenever tryptophan is required by the cell, this particular operon should be switched on, because tryptophan is an amino acid. So in many enzymes or in many proteins, tryptophan will be required by the bacterial cell. Whenever tryptophan, the amino acid, is required, this particular pathway should be switched on. That means the repressor protein should be activated when the bacterial cells require tryptophan. The repressor protein is unable to bind to the operator by itself. That means the repressor is different from that of lac operon, because the repressor protein in the lac operon, it does not require anyone to bind to the operator. When it is produced, it directly moves and binds to the operator. But whenever lactose binds to it, in lac operon, it cannot bind to the operator. But the situation is opposite in the case with the repressor protein in tryptophan operon. That is, the repressor protein can bind to the tryptophan operon or the operator of the tryptophan operon only in the presence of tryptophan, which is considered to be the co-repressor. When only binding with the, that is the repressor protein bind to the operator only when it is complexed with the co-repressor. So here also we are going to take the two situations, that is the situation when tryptophan is present and when the situation is absent. So you can see the tryptophan operon, the diagrammatic sketch of tryptophan operon. So you can see the regulatory gene which is seen downstream of the promoter, followed by the promoter, then you have the operator gene and the 5 enzymes. Here the name of the enzymes, you need not study the name of the 5 enzymes. So there are 5 enzymes which are responsible for the synthesis of tryptophan. So now we are going to see how this is being regulated, how this is switched on and how this is switched off. First we are going to take the situation where tryptophan is absent inside the cell. That means when tryptophan is absent, it should be synthesized. Should be synthesized means your operon should be switched on. So what happens here is, so when tryptophan is absent, as we said earlier, only when tryptophan is present it can bind to the operator. When it is absent, the repressor protein, even though repressor protein will be produced, it cannot bind to the operator. RNA polymerase is free, it transcribes the structural genes which is required for tryptophan biosynthesis. As a result, tryptophan will be synthesized by the bacterial cell. Take the second situation when tryptophan is present. When tryptophan is present, it binds, being the co-repressor, it binds with the repressor protein, forms a complex, binds to the operator region, and it is inhibiting the transcription of the gene, and hence tryptophan will be, the production of tryptophan will be inhibited. So this is also called as a repressible operon. The other one was an inducible operon. This is a repressible operon because the end product of the biosynthetic pathway, as we said earlier, the enzymes in this biosynthetic pathway is causing the synthesis of tryptophan. So when this particular process, of when all these enzymes act, the end product is tryptophan. So when the end product of the biosynthetic pathway, in this case tryptophan, is repressing the operon. That means the operon was responsible for producing that particular end product, tryptophan. That end product is now inhibiting the operon by complexing with the repressor, and such operons are referred to as repressible operon, where the end product of the particular metabolic pathway or the operon is inhibiting the same operon. That is called as a repressible operon. So here what happens is, whenever tryptophan is absent, as we said earlier, tryptophan, the biosynthesis occurs, the amount of tryptophan increases. That is according to the need of the bacteria. Tryptophan will be used for many production of many enzymes, many proteins, etc. The amino acid will be used, and whenever the production exceeds, that means it will be producing the mRNAs, the biosynthetic pathway will move on, more and more tryptophan will be produced. Whenever tryptophan production exceeds than the bacterial cell needed, now these excess tryptophan will act as the co-repressor. That goes and binds to the repressor. Now it represses the operon. That means whenever tryptophan production exceeds than the bacterial cell requirement, this will be switched off. So this is how this particular tryptophan operon works. So as we said, in the lac operon, there was a positive inducer, the CAMP, and also glucose, which was showing something which is called as glucose effect, etc. Here in tryptophan operon, we are having something different from that. This is also an additional regulation in tryptophan operon, and that is called as an attenuation. So this is for tryptophan operon. Attenuation is the operon which is controlled by specific regions of the mRNA. When the operon is controlled by specific regions of the mRNA, that is called as an attenuation. So here, in the presence of what happens here is high concentration of tryptophan, RNA polymerase ceases transcription shortly after initiation, and initiation in a region which is called as a leader sequence. So what happens is, as we said earlier, when tryptophan is absent, tryptophan operon is on, it is synthesizing tryptophan, etc. It moves on and on. So whenever the amount of tryptophan is exceeding, it is acting as a repressor. So we have to assume that our operon is switched on, the polymerase is moving. So whenever this switch off message is given to the operon, or when the co-repressor and repressor moves and bind to the operator region, what happens is our RNA polymerase might have moved forward than the initiation one. Even though it has moved forward, you can inhibit the process. That is what is called as attenuation. So we here what happens is, after there is a particular sequence after the initiation point which is called as a leader sequence. If tryptophan concentration is low, transcription does not terminate until the entire operon is transcribed. It will move on, transcribe the gene, which happens normal. The mechanism of this attenuation links, or attenuation, occurs due to 2 RNA secondary structure. So immediately after transcription, the RNA from the leader sequence, that means the leader sequence is in the mRNA. It is seen in the gene as well as the sequence can be seen in the mRNA also. So immediately after transcription, the RNA of the leader sequence, it can assume 2 shapes. One shape is a termination signal. That means, as I told you, whenever this switch off signal or switch off signal comes at the operator, but assume that the RNA polymerase have moved on, it has synthesized the leader sequence, etc., or it is synthesizing the leader sequence. Whenever this particular message of binding of our repressor and co-repressor reaches the operator, what happens is that the leader sequence will assume a secondary structure which will be terminating, which will be acting as a terminator signal, and it will terminate your RNA polymerase from moving along. But in an alternate structure, that means, as we said earlier, it assumes 2 structures. One is when the particular operon should be inhibited. The other when the operon should be stimulated. When the operon should be stimulated, the leader sequence does not assume a terminator signal. It assumes a signal which favors transcription of the whole operon. So this is what is called as attenuation. So here this is called as attenuation because as we said earlier, the attenuation means the operon is controlled by the specific region in the mRNA. Here the region is leader sequence. Leader sequence assumes two structures. One is a termination signal and the other one is an initiator signal. Termination signal occurs when tryptophan concentration increases in the cell than the bacterial cell requirement. So that particular signal causes the RNA polymerase to terminate transcription before transcribing the complete operon. But in the other case, what happens is the alternate secondary structure, it will initiate or activate your RNA polymerase to transcribe the entire operon and continue producing the enzymes which are required for the synthesis of tryptophan. So this is all about tryptophan as well as the lac operon. Coming to eukaryotic gene regulation. So eukaryotic gene regulation, this is in your syllabus, it is mentioned that there is only a brief account of this particular gene regulation in eukaryotes, but you can expect a 5 marks question from this. So there are different levels by which eukaryotic gene regulation happens. This is entirely different from the case with prokaryotes because you do not have an operon here. Genes are separate in chromosomes and hence controlling a single gene, it will be switching on and off that particular gene. It is not a string of genes as we saw in operons. So here we are going to see what or at what levels or what are the strategies that a eukaryotic cell takes for regulating the genes in a eukaryotic cell. So first is chromatin modification. So that is the first level. Though at different levels you can regulate a gene. That means the first level is you are going to inhibit the gene from transcribing. That means you do not allow the gene to transcribe. That is the first level, which is called as chromatin modification. That means either you can activate it or you can inhibit it at the level of DNA itself. That means you can switch on and off that particular transcription at the level of genes. That is what is called as chromatin modification. Second level is the gene is going, or the gene is now ready for transcription. So you are going to decide whether the cell is going to decide whether transcription should happen or not. So that is at that level. That means the mRNA should be produced or not. So it can produce the mRNA, you will have the activators, you can repress it, you will have the repressors, etc. That is the transcription level to decide whether the mRNA should be produced or not. Second is after the production of the mRNA also you can regulate it, which is called as post-transcriptional. You can degrade the DNA, you can cause the more and more you can not the DNA, you can degrade the mRNA. You can produce more mRNAs if you need, depending upon the situation. That is post-transcriptional control. The next is you can also regulate it at the other point, which is translational control. That means you can regulate whether that particular mRNA should be translated or not. The last thing, if at all that protein is produced and the cell now does not want that protein. So all these levels are over. That is chromatin level, it was okay, it was transcribed, mRNA was produced, it was translated, the protein was produced, but suddenly the particular need for the cell for that particular protein is now not in action, or that particular enzyme is not needed now. So at that point also, even though all these processes have happened, at that particular protein, a protein is produced now. If the cell does not want that protein can be degraded. That is what is called as post-translational control. So at these different levels in eukaryotes, a gene or a particular gene can be, or a gene or the particular product of the gene can be regulated. So first is chromatin modification. That is the first decision whether that particular gene is to be transcribed or not. So as you all know, in a eukaryotic gene, it is usually found in a solenoid structure. That means it is compactly packed. The genes are not seen, or it cannot be seen as free as double-stranded DNA or as a chromatin. Whenever the DNA is exposed, your DNA polymerase or an RNA polymerase, surely it will come and bind. So only for constitutively expressed genes that particular DNA will be seen as something, the terminology which is called as naked DNA. Naked DNA means it is a double-stranded DNA which is not packed inside the solenoid. When a naked DNA is exposed, it becomes a site for or a region for RNA polymerase to bind. So now the chromatin modification is a decision whether your gene should be formed a naked DNA, or it should be inhibited from forming an mRNA. So there are different processes for this: acetylations, methylations, phosphorylation, etc. Usually acetylation and methylation or acetylation usually causes a naked DNA to show up. That means it favors transcription. Methylation and phosphorylation, what methylation and phosphorylation does, that means whether it is whether it is initiating or activating transcription or inhibiting transcription, it depends upon the gene. For some genes, methylation causes repression of transcription. That means the DNA will not show up. It will be present in the solenoid, compacted. It will be saved from the RNA polymerase. But in some cases, you can see methylation is activating transcription. Phosphorylation with the same thing. So it depends upon the gene. Acetylation, usually acetylation, that LYS is nothing but lysine. Acetyl groups are usually added to lysine residues in the histone protein. So these chromatin modification means you cannot modify the DNA. So mind you, when you write the answer, if you remember the term chromatin modification, don't go and write it is modification of the DNA. DNA is not modified. If a DNA is modified, the complete gene will be cramped up. So what here happens is, here the modification of chromatin means you are supposed to either tighten your DNA or loosen your DNA. Loosen your DNA means DNA will show up and RNA polymerase will do transcription. The otherwise, you are going to tighten the DNA. As a result, no transcription occurs. Tighten the DNA means you are not allowing the histones to move out of the DNA. Acetylation of histone protein, all this process, acetylation, methylation, phosphorylation, all these are the processes that is being done on the histone proteins. So when in the case of, for an example, in the case of acetylation, when lysine residues of lysine, which is an amino acid which is present in the protein which is histone, when the lysine residues in the histone proteins are acetylated, these histone proteins lack its affinity for DNA. Histone proteins move. When histone protein move, the DNA will show up as double-stranded DNA. Then transcription occurs. So mind you, chromatin modification is not modification of the DNA. It is modification of the histone proteins to either remain as solenoid or to relieve the DNA for transcription to occur. So that is what is happening in chromatin modification. Next is the second level of control, which is transcriptional control. That means whenever the DNA has showed up, now the decision is whether that particular DNA is to be transcribed or not. So for that control, you have two elements, proximal control elements and distal control elements. So proximal control element is nothing but the promoter, which we discussed earlier. Tata box is nothing but the name of a eukaryotic promoter. So we discussed earlier how the promoter is acting. So promoter is the region where your RNA polymerase, the same thing happens in eukaryotes. It is the region where RNA polymerase binds. If a repressor or something is present in downstream of the promoter region, it represses the other thing. It does not activate transcription. The two terminologies, proximal and distal. Proximal means near, distal means far. So proximal means proximal control elements means these are the sequences which are seen very near to the gene to be transcribed. So promoter, obviously it is very closer, or which is seen just upstream of the gene to be transcribed. And there are a second set of control elements, which is the DNA sequence itself, which is seen far away from the gene which is going to be transcribed. And those control elements which are seen far away from the gene, they are called as distal control elements. The examples are enhancers and silencers. Okay, enhancer is a DNA element. So here I have put it for your easiness. The distal control elements are the